Works. Firstly, I just wanted to say thank you very much for inviting me, and also just to say that of everybody in the room, I probably know the least about well, archaeology, full stop, but Norman archaeology certainly in the specific. But I've been intrigued by just in the last hour and a half of especially in our breakout group, of the number of issues and themes that are cross-cutting between a lot of what we do in our lab and a lot of what's happening here. And it's intellectually highly stimulating to know that there's a lot of higher level questions here that are being like explicitly queried, and hopefully will result in a much more sophisticated or nuanced understanding of everything that we're talking about here. We had a big debate about whether you can use nuance or sophistication at all in academia, but so I will run through this very quickly and happy to answer any questions. I'm aware that many of you are not geneticists, but what I wanted to demonstrate with this was a case study to show how genetics can be integrated alongside both linguistic evidence and archaeological evidence to come to a whole new understanding of something and give us a new perspective and a much better and a clearer narrative with a kind of mind-blowing connection between these things. So very generally, what we do in our lab, I try and, if you could explain what you can do in 2 images, this is us. We take old bones, we put them on machines that go ping. That's pretty much what we do. This is actually an older machine. I like the lovely blue light back and forth, like a HAL or something. I'm sorry, we can't do that. Yes, you can. These are cow bones, of course. We work on a variety of different, mostly domestic animals, but some wild ones as well. And I just threw in a couple of slides very quickly because a lot of the bigger questions that we were asking in our group about the Norman invasion and what is it, and Alex's introductory talk all about, well, what is it? Are there multiple? Is it an event? Is it a process? It's exactly a paper that we have that we've submitted and is just about accepted. We submitted the final version of it yesterday, and it's all about rabbits, the domestication of rabbits. And what we realize is that there is no such thing as domestication of rabbits. So this paper, so I have a bad idea to read text, but I'm just going to read this to you. This is from the paper itself. All the evidence that we've compiled in this, looking at genetics and archaeology and linguistics, demonstrates that, or reveals how frequently the domestication process itself is misconstrued as a discrete event. We have this all the time. People, you go to conferences, even for people who should know better, talk about domestication events all the time. It just fills me with dread. Instead, the combination of the methodological and semantic factors highlighted in this study suggests that a precise domestication date does not exist. So every time you reach for when were dogs domesticated, when were rabbits domesticated, the closer you get to what's supposed to be there as a reified, actual, hard, black and white event, just vanishes. It's sand running through your fingers. It's not there at all. It all comes back to your definitions, and your definitions are based upon our current understanding or our current relationship to domestic animals, and that wasn't the case X number of 100 or 1,000 years ago, and so therefore it all is kind of meaningless. Which sounds horrible, but isn't, because it's kind of fun, because at least we're realizing the limitations of this. So a bit more specifically, the domestication of rabbits specifically was the result of a continuous dynamic process that reflects gradual shifts in the nature and intensity of the relationship between humans and other species. It sounds very much like the Norman or any sort of invasion that we're talking about. And lastly, to obtain a satisfying rabbit domestication narrative, again a word that came up a lot, we need to view domestication and its associated biological changes as a process that occurs along a continuum. And so that's this whole issue of, if you have a continuum, if you try and reify any stage along that continuum as a black and white, as a dichotomy, you're necessarily going to fail, or at least that's going to be entirely predicated on your definition, which itself is predicated on a series of assumptions you're making that are likely to be untrue. So how do we appreciate that continuum for what it is without breaking it into a dichotomous have, which then kind of falls apart and is not very satisfying for anybody. So I wanted to give you a specific example of how we're trying to overcome that. So that's the theory. This is the specific case example, and it's involving chickens. And Naomi is sort of the effective PI as it was the AHRC chicken project, which finished up just a little while ago, although we have a ton more data that we're still analyzing and lots more papers that are coming out. And these are, what I like about this is that these are modern chickens. These are modern fancy breed chickens, which even 100 or more years ago simply did not exist. We have gone absolutely insane in selecting chickens and dogs and rabbits and all kinds of things for increasingly bizarre morphological novelties that would be unrecognizable to our Norman forebears, as it were, because these things in many ways are simply not chickens. And if we look at these and we expect to find these in the archaeological record, we're going to be, again, the sand's going to be flowing through our fingers. So the story involves it starts in about 2010 when I did a postdoc in Sweden, and my PI there is just after I left, so I missed out being on the paper, but published a paper where he said that they'd found a gene when they looked at the differences between domestic chickens and wild red jungle fowl, and it was a gene called TSHR, which stands for thyroid stimulating hormone receptor, which is, everybody's got an endocrine system, we all have thyroid, and these chickens are no different. And what they found was that this gene TSHR was what they called a domestication gene. This has been found in many plants, and nobody found one in animals. And they said where all the domestic chickens carry a mutant allele, which you do not find in the wild in red jungle fowl, even in zoos, like for a very small proportion. So, and it's a reasonable assumption to make that if every domestic bird has this variant and none of the wild ones do, that it must have been selected for very early on in the domestication process. You could even say that there was a domestication event when people started selecting for birds that then acquired this trait and allowed them to proliferate within human societies. So what we did a couple of years later was we took this gene and we said, well, if that's the case, let's look for it in the ancient birds to see in Europe, and let's see if it was 100% fixed by the time that domestic birds get to Europe, which of course is thousands of kilometers away from red jungle fowl. And so if it was very much happening at the origins of chicken domestication, by the time they get to Europe, everybody should have it. So this is what it should look like. Each block here is an individual ancient domestic bird that we looked at going back to about 200 BC all the way up to about 800 AD across Europe. It's about 80 individuals here. This is what it should like if red is what's called the sweep allele or the domesticated version of the gene. Instead, when we type them all using ancient DNA, again, taking our bones, putting it on the machine and having it give us some sequences back, it looked like this, which was rather, I mean, for me, it was terribly satisfying because I didn't believe this narrative in the first place. I was like, it's not true. And so, look at that. What do you know? It's about 40% of these birds. In fact, very few of these birds were what are called had both alleles. So because everybody has two chromosomes, and so you have two copies of this gene, and a lot more of the birds had two copies of the wild type gene than they did the domestic one, even though these are very clearly coming from domestic birds. So what does that suggest then about how important this was at the origins of chicken domestication? If by the time you get to Europe, most of your birds still have the wild type allele, when was this gene actually being selected? So we took this study a little bit further, and what we said we want to know is the starting time of that selection, if it wasn't part of the original domestication event, as it were. What was the starting frequency when that selection started to increase, because now all domestic birds have 100%? And what was the strength of that selection in a sort of very relative way? Was this happening ridiculously quickly, or was it happening a little bit more gradually? So we used what is this ridiculous likelihood framework thing, which I know very little about, but my PhD student knows a whole lot about, which is the fun thing about running a lab. You bring in people who know a hell of a lot more than you do, and you just take them at face value. You're like, yes, that makes a lot of sense. Let's go and run that one. And what's fun about it then is you can look at these three parameters, the values for them, and you can compare them against the actual empirical data that we generated. We could say, then what are the values that fit what we can see on the curve? And so what we do is we say we have a starting time for selection on one axis, the year of the starting proportion of the allele frequency, and the year of when it started kicking off on that axis, and it creates what's called a likelihood curve, where the blue is the less frequent combination of those parameters, and the warmer the colors, the more likely it is. And the first thing that's fun about this is that we're studying chickens, and it gave us an egg. We're like, hey, perfect. The other thing is that when you run an analysis like this, it doesn't have to look like this. You could have blue everywhere, which would be no combination of any two parameters or any more likely, or you could have red everywhere, which said everything's equally likely, but probably pretty likely, in which case you have no way of picking out which selection or what proportions of these two parameters were actually more valid. And when we look at the hottest point, so imagine this is a landscape and the red is the top of a peak. Because we have a nice top peak there, high above everything else, we can say that it was about a 40% selection pressure, or 40% starting frequency rather, of this gene. And about 1000 AD, that's when it started rocketing up. And this just emerged out of the data. So when we plot it like this, these are the years on the bottom, this is the proportion, you can see all the way at the very top right. That's where we are today. 100% of domestic birds have this very easily identified allele that differentiates them from wild birds. And about 1000 AD, that's when it starts climbing. It climbs very quickly relative to any other gene that we know is under selection. So when we got this result, we were all very impressed. Now, most of what I do is work on early stages of animal domestication, and my knowledge of even the term AD after a year starts to throw me a little bit. I'm like, what is AD even? I don't even know what happens after, everything is way BC or BP even for me. And this is the PhD student who was working on it. And so this was in the notes when we started writing up the manuscript. She put this in the notes. She put, we find that the most likely starting time for selection is 920 AD with a confidence interval, which is older than selected by Flink et al., which is the original paper. Then she puts as a note to me, what changed in how chickens were managed? Comments from Gregor Stroke, archaeologist. Now there's an important first point, which I should point out. Though I reside in an archaeology department down south, she was clearly differentiating me from all archaeologists because she knows that I'm not an archaeologist, which is fair enough. But the other point I like about this is that it demonstrates that these were numbers that she pulled out as a molecular population geneticist, having no knowledge of archaeology or history or even chickens for that matter. This was just a model system for her where we were working out this, and she was doing it more from a methodological framework, working out that likelihood function to see how we could get to the number. The number itself was meaningless, and she didn't know. So, of course, what I do, I called Naomi and I said, because I knew that Naomi knew a lot more about this than I do, and I said, Naomi, does 1000 AD mean anything to you in terms of why people might be changing the way they're interacting with the chickens? And she said, are you kidding? Really 1000? I said, yeah, does it mean anything? She's like, have you read my thesis? No, I have not read your thesis. And she said, read my thesis. So we then together what Naomi did was pull a whole bunch of different sites together and look at the changing proportion of chicken remains against all other faunal remains in large numbers of archaeological assemblages across the UK. And what she found was that between the 5th and the mid-9th, the mid-9th and mid-11th, chickens represent about 6% of the overall faunal proportion in these assemblages. But then, starting at about 1000 AD or so, between the mid-11th and mid-12th, it rockets up. It about doubles, suggesting that people are putting a lot more effort into raising, possibly eating, or at least managing chickens in much larger flocks than they had been doing beforehand. So we then took all of Naomi's data, put some nice colors on it for the paper to show this significant increase. And then we spoke to Joris Peters and we said, Joris, is this happening not just out in England, but outside of England? Can you look at all the German sites? So his PhD student then surveyed as many German sites as Naomi had in England, and we see exactly the same thing in Germany. Maybe not quite as pronounced, but you still get a significant rise, again, at exactly that same time set. So this is not just an England-specific thing, this is happening European-wide. And so then what's happening at 1000 AD? Well, this is I keep saying Pope Benedict, but it's not Pope Benedict, right? Yeah. So this is Benedict, alive from 480 to 543, and about 500 years after that, there was a return to the Benedictine reform, it was called, and all of you know this much better than I do. And there was this return to this monastic living, or at least a big push on it. This was sort of like the religious right taking over again, where they were saying, look, people need to be fasting much more deliberately than they are right now, not kind of playing fast and loose with the whole idea, so that four-legged, during fasting times, you cannot eat four-legged animals, but chickens and eggs are okay. So because this became much more enforceable, at least that's the idea, people then were like, well, okay, if we can't, there's 120 days a year or so that they're not allowed to eat four-legged animals. Chickens are a nice viable alternative for the meat, so that starts to increase significantly, and all this is happening at about 1000 AD as well. So that suggests that when we take the genetics outside of the context of anything else, we look at the archaeology outside of the context of anything else, and we look at the actual historical records, what's happening, all three of these things are pointing to about 1000 AD. And that's suggesting that there is something significant happening in the way in which people are interacting with one of their domestic animals, which they had already. But then what happens is that they start, because of that shift in the way they're interacting with the animal, they change, however deliberately or not, they change the selection pressures, which then starts altering the frequency of a gene that was already present, but not at very high frequency, driving it to fixation. And then because of the presentist fallacy, we look at modern animals and we assume that this is the way it's always been. So then we take that proportion, we reflect all the way back to the beginning and then demand that this was a gene that was part of the domestication process itself, when in fact it's only 1000 years old. So what I like about this is that it demonstrates the dynamism with the changing ways in which people and even animals that they've had around them for a long time can rapidly shift and then alter to the same degree of those first chickens that I showed you, which didn't exist even as long as 100 years ago. And now we have these crazy fluffy things that you can buy calendars of and impress all of your friends if you have some in your backyard. So if we're thinking about origins, and I think this is what a lot of the Norman conquest is about as well, is that and this is the fun thing for the press release, but this is the first time perhaps that chickens are being thought of as fast food. Before that, people interacting with a different way. We're not saying they're not eating them, but they're certainly not eating them nearly as much as other domestic animals. But now you start almost harvesting them on a much larger scale than you had before. And our current relationship with chickens, which is vastly different than the first 3,000 or 4,000 years that we were ever around them, is now so changed that it's hard for us to even reflect on what it must have been like at the very beginning, used in ritual practices or many, many other things. But the nice thing about this is that the Catholic Church then allowed chickens to be eaten guilt-free, and which is what we do now, because we don't even see the chickens, only except for as tiny bits and pieces of parts in supermarkets under foil wrapping and everything else. So I think that what's nice for me about that is that it demonstrates, and this is something I put on one of the post-it notes, is that when we're thinking about these things, especially in a historical context, when we have historical records and we have iconography and we have coins and we have all these things, that we link that with the archaeological record and then use the genetics to get us a degree of resolution that would really not be able to be there without having the very fine scale sequences, but then that in conjunction with, we know, very large scale patterns of zoo archaeology as well, I think that can start to reveal patterns to us that start to make, I hate to say a lot more sense because that means that it only is true because it makes sense. But what I mean is that it's a much more satisfying way of appreciating it because of the intersection of all these different things. And when they start pointing in the same direction, you feel like you're on some kind of track, which is telling you something interesting. Anyway, thank you very much.